Welcome. In the last session, I discussed up to concept of dominance. Now in this session, I'm going to discuss a co-dominance, and thereafter I'll also discuss about the starch synthesis in pea seeds. So let's begin this session with co-dominance. Right. So here, whatever the process we have seen earlier, so in all those process, that is a monohybrid cross, dihybrid cross, or incomplete dominance, in all those crosses, we have seen that F1 generation resembles either of the parents, but not both. But in case of codominance, we are going to come across a situation where the F1 generation will resemble both the parents. Okay, so that is in codominance, the F1 generation resembles both the parents. Okay, so here we'll take an example and understand how the F1 generation resembles both the parents. So here example is ABO blood groups in humans is a very good example to understand codominance. So here the ABO blood groups actually in humans. Okay, so there are four phenotypes. So that is A blood group, then second one is B blood group, then the third one is AB blood group, and fourth one is O blood group. Okay, so these are four phenotypes that we observe in case of human beings, and these blood groups in human beings is controlled by a single gene. Okay, so all these are controlled by a single gene. Okay, so that single gene is I. Okay, so here the I stands for isohemoagglutinogen. So this gene I is responsible for synthesis of a antigen on the surface of plasma membrane of RBC. Right, so it is the gene I will be responsible for the production of a certain type of antigen on the cell membrane of RBC. So this is an RBC, and it's the plasma membrane. So here protruding on the plasma membrane, we find some special type of structures, and these are called sugars. So these are a type of sugars. Now depending on the type of sugar, we are going to classify whether it is A blood group or B blood group, AB blood group, or O blood group, right? So here all these blood groups are controlled by a single gene I, and this gene I is responsible for production of certain types of sugars, which will be seen protruding on the plasma membrane of RBCs of human beings, right? So here these are, I just mentioned, sugar polymers. So they are sugar polymers, which will be seen protruding on the plasma membrane of RBC, right? So now this gene has alleles. So we know, right? So the gene I, So the gene I has, okay, three alleles. So that will be IA, IB, and IO, or I'll mention it a small I. Okay, so that will be the recessive one. So here there are three alleles for this. Okay, so three alleles. But we know that in a diploid organism, there will be only two copies of each gene as a pair of alleles. So in a diploid organism, so in the last session, I told this, in a diploid organism, there will be two copies of each gene as a pair of alleles. So, but how here three alleles have come up? So this I'm talking about in human population, not in one individual. Okay, so if you take in one individual, you will find out of these three only two. Okay, so if I'm talking about a particular person, say in a X person, okay, so in a X person, out of these three alleles, only two alleles will be present, maybe like IA and small I, but not all the three. So here these three alleles are actually right in population. So when I'm considering the population, okay, so we know population is a group of individuals belonging to same species. Group of individuals belonging to same species, okay, is a population. So here the multiple, so this is what we call the multiple alleles, and multiple alleles are found in population, not in a single individual, right? So at an instance, only two out of these three alleles will be present in a person. Okay, that's what, right? So because humans are not triploid, so we are diploid organisms, so hence we will have, okay, the genes as a pair of alleles, that is only two, okay, copies will be present, right? So the gene I is capable of producing, okay, so this sugar polymer. So if it is able to produce this sugar polymer, say it's going to produce a sugar polymer, then the person is said to have, okay, said to have a blood group. So here, so a sugar polymer, so then in this case the person is having a blood group. So you have to keep in mind, okay, this sugar polymer is the one that we consider as antigen, is antigen. So antigens will be present on the membranes of cells, and here we will see the antigens present on the RBC. I'm considering, of course, all the cells of our body will be having antigens on their cell membranes, but here especially I'm talking about RBCs. Now the type of sugar polymer that is present on the cell membrane of RBC will decide, okay, the person's blood group, and that will be decided by the gene I. Okay, so if the gene I produces this A, okay, sugar polymer, then of course I will say in simple words as antigen A. So if this one, I'll just mention, produces antigen A. So if it is producing antigen A, then the person's blood group will be A. And this one, it produces antigen B. So it produces antigen B. So if it is the case, then the person's blood group will be B. And okay, no production of antigen. So antigen is not produced. So if antigen is not produced, then it is said to be O blood group. So here on the surface of RBC, we don't find any, okay, sugar polymers. So if no sugar polymers are present on the plasma membrane of RBC, so here no antigens, no antigens. In this case, this will be the O blood group. So the person's blood group will be O. So if you find, say here, I'm going to just mention here, so this is antigen A. So here this is antigen A is present. So then the person's blood group will be, A antigen I said, and if you see, B antigen also. So this is B antigen protruding on the plasma membrane. So this is antigen B. So now a person is having, right, both antigen A and antigen B on his RBC membranes. Then his blood group will be the AB blood group. Will be AB blood group, right? So I hope this part is clear. Now here this gene, that is, or the allele IA is dominant. So this is a dominant one, and this is also dominant, so while this one is recessive, so that is the reason why I have mentioned with the small I. So this is recessive, and here when antigen A and antigen B are present on the RBC membrane, or the gene for this antigen A and antigen B is present, then both will get expressed, right? As a result, the person's blood group will become AB. So here you have to keep in mind that the antigen A and antigen B are codominant. So they are codominant. So here in our earlier processes, we have seen that out of two alleles, only one used to get expressed and one used to hide. And the hidden one is what we used to call recessive, and the one which is expressed, we called it dominant, right? But here in this case, we have come across, of course, when Mendel was doing experiments, he did not come across such a situation. And even in case of incomplete dominance that was discovered by Carl Correns, so Mendel also did not come to know about incomplete dominance. That is what I have given the title, deviations from Mendelian laws. So here it produces the gene, that is I. If it produces antigen A, then the person's blood group is A blood group. And I said if the person's RBCs are having antigen B, then his blood group is a B blood group. And if no antigens are present, either A is not present, B is not present, so either of them are not present, then the condition becomes O blood group. Right? So, and A, B are dominant. So when they are present together, both will get expressed. So that's, I'll just mention. So A is co-dominant to B, and these two are dominant over O blood group. Are dominant over O blood group. So just this is not any mathematical equation here. I simply mean the A blood group and B blood group are co-dominant over the O blood group, right? So these two are co-dominant, and this is the recessive blood group, right? So here I already told you that there are four phenotypes. Okay, that is A B and O. Okay, phenotypes. So there are four phenotypes. A, the next one is B and AB and okay, O. So these are the four phenotypes, but there are six genotypes possible. Okay, six genotypes. So let's see what those six genotypes. One, so IA. So here in this condition, okay, the person's blood group will be A blood group. So it is A blood group. Okay, so we know that, okay, this IA has come from one of the parent, and this one is another gene that has come from another parent. So say suppose this has come from the male parent and this has come from the female parent. So that is one from the father and from the mother. Now the child's blood group will be A blood group. Okay, so this is the genotype. So next we'll see in case of IA and small I. So here this recessive gene he inherited from one parent, and this IA inherited from, okay, and another parent. So in this case also, because we know this is dominant and this is recessive, when dominant and recessive present, we know it is the dominant that gets expressed, and in this case also the person's blood group will be A blood group. The next one, IB. So we know if both are same, then this is a homozygous condition, and this is a heterozygous condition. Okay. Now again, in homozygous condition, so if he receives IB and IB, okay, from the parents, then this will be the B blood group. Then, okay, and another one, so in heterozygous, so one gene, okay, that is the dominant gene IB, have received from one parent, and recessive gene from another parent. Then in this case also the person's blood group will be B blood group. And then in the next situation, say suppose if a person received one gene, that is IA from the, okay, father, and IB from the mother, then in this case the person's blood group is AB blood group. The person's blood group is AB. And lastly, if a person receives both recessive genes, okay, then the person's blood group is O blood group. The person's blood group is O. Okay, so you can see here there are 6 genotypes and 4 phenotypes in this case, right? So here O actually means absence of antigens on its okay, cell membrane. I mean RBC's cell membrane. Now let us take an example and understand how inheritance of blood groups is seen. Okay, say suppose a person with A blood group. So a person with A blood group, okay, say the person is heterozygous. The person is heterozygous. So a person with A blood group marries, okay, a woman with B blood group. So in this case, woman is also heterozygous. Okay. Now then find out the types of blood groups in their, okay, children or in their offspring. So if they have, okay, children, then what type of blood groups we will be seeing, okay, in their children. So let's see, right? So here first what he mentioned, a person with A blood group. So I'll just mention here instead of this, a a man with A blood group marries a woman with the B blood group. Both are heterozygous conditions. So let's see here, A blood group. So here they have mentioned that it's a heterozygous. So A blood group, heterozygous condition. So it means that I need to take, okay, capital I A and small I. So this is the heterozygous condition. Okay, now marries a woman with the B blood group. So woman with the B blood group. So in this case, woman is also heterozygous. Okay, so now I need to take capital IB and small I. Right. So here parents, okay, 2N. Now during the formation of gametes, we know that these are two different alleles and they get separated, okay, in a 50-50, right? So 50% of gametes will have IA and 50% of gametes will be having small I. So this is IA and this is small I. Okay, so I'll just mention here, so 50% of gametes and 50% of gametes are having recessive, okay, allele. And in this case also same, IB, so 50% of the gametes, and small I, so 50% of the gametes. So gametes N. Okay, now let's see, if the gamete having IA and the gamete having IB, so if these two, okay, get, okay, I mean if they are fused or fertilized, then it results in, okay, the formation of okay, IA IB. So here the A blood group person is, okay, male and this is female. So I'll mention, okay, 50% of sperms and this is also 50% of, okay, just mention 50% of sperms and 50% of sperms. So this is 50% of X and this is also, right. So this one, if it comes in contact with this one, of course, if you feel that this is okay, a little bit confusing, so we can put it in Punnett square and okay, see the possibilities. Okay, there is that can also be done. So here IA, so if this sperm having IA, okay, fertilizes the egg having IB, then it will be IA IB and the born baby will be having AB blood group, will be having AB blood group. Okay, and if this IA, right, comes in contact, means the sperm having IA comes in contact with the egg having, okay, small I, then it will be, okay, IA small I. In this case the child's blood group will be A. And here the egg having IB, if it comes in contact with the sperm having, okay, small I, then it will be IB small I. Then, okay, the born baby will be having, okay, B blood group. And if this and this comes, so small I, then the blood group will be O. So this is F1 generation or offspring. Okay, so I will just, right. So now we understand that when a person who is heterozygous with the A blood group and heterozygous B blood group, okay, woman, when, okay, when they marry, there is a possibility that their children, okay, will have all the four types of blood groups. So here the chance of their baby having AB blood group is 25%, is 25%, and the baby, okay, to have A blood group is again 25% of chance, and the baby having B blood group 25, and this is also 25. So there is a chance of 25% of each blood group, okay, to the offspring born, okay, to this couple, right? So this is how we can come to know the inheritance of blood groups in human beings, right? So I'll take one more example to understand better, right? So here a man having O blood group, a man having O blood group marries a woman having okay, B blood group, a woman having B blood group. So in this case, I'm not mentioning whether we know in case of O blood group it will be always homozygous, okay, whereas in case of B blood group I'm not going to mention whether it is heterozygous or homozygous, okay. So here, so what will be the type of blood groups in their offspring? So what will be the type of blood groups? So what will be the type of blood groups in, okay, in their offsprings? So let's see. Already in case of male, I said it is O blood group, and we know O blood group will get expressed, okay, in homozygous condition, right? So that will be small I, small I, and now in case of woman is having B blood group, having B blood group. So here as I did not mention whether it is homozygous or heterozygous, first we will see with the homozygous condition. So IB, so parents, okay, 2N. Now during the formation of gametes, the male will all the time produce, okay, the sperms having only small I, 100% of sperms will be having this, so 100% of sperms, right? And in this case the X will also have only IB, so this will be 100% of X, okay, so gametes N. Now the sperm and egg fertilization, and that results in the, okay, formation of the, okay, young one, it will be IB and small I, okay, so offspring, or I can use another word, progeny, right? And in this case it will be B blood group, okay. Now if this couple is going to have 10 children, all the 10 will be having only B blood group, or if they are going to have 100 children, then all of them will be having only B blood group, never ever will be the O blood group gets expressed, right? And we'll take, this is the condition, okay, in if the woman is, okay, homozygous, right? So if none of their children, if none of their children have O blood group, then we can come to a conclusion that the mother is homozygous, right? And if we see a O blood group in the offspring, then we can come to know that the mother is heterozygous. So that's how we will see. So here I have taken in homozygous condition. Now I'm going to take in heterozygous condition. So small I, parents, okay, diploid, and this will be as usual, okay, 100% of sperms. But here, okay, 50% of eggs will be carrying this IB, and small I in 50% of eggs. So gametes N. Okay, so now this one and this one, it will be IB and small I. Then okay, the young one's blood group will be B blood group. And if this one and this one comes, small I, O blood group, right? So here it indicates that there is a 50-50 chance. So this is 50% of their offspring will have, okay, B blood group, and this will be 50%. Okay, if the mother is heterozygous, right? So that's about inheritance of blood groups in human beings. I hope it's clear, right? So now we'll move on to the next topic, starch synthesis in pea seeds. So the next one we'll see about starch synthesis in, right, so pea seeds, right? So here starch synthesis in pea seeds is controlled by a single gene. So starch synthesis in pea seeds is controlled by a single gene, and this single gene has two alleles. So that is capital B and small b. Okay, so these two are alleles. So the starch synthesis in the pea seeds is controlled by a single gene, and this single gene has, okay, two alleles. So now let us see when the genotype. Okay, so let's see a tabular form. So here, size of starch grain, and okay, shape of the seed. Okay, so now here when the genotype is capital B, so when the genotype is capital B, so when it inherits, okay, both genes, okay, dominant genes, okay, so here with this we can understand capital B indicates it's dominant, and this one indicates recessive, right? So when the plant inherits both dominant genes, then it will produce large starch grains. It produces large starch grains, and the presence of this large starch grains makes the shape of the seed to be round in, okay, so round in shape. Then coming to in the next condition, when it inherits, okay, both the residue, that is small b and small b, then in this condition it produces smaller starch grains, and this will result in having wrinkled shape of the seed. So it will have wrinkled shape of the seed. Okay, now here in a third condition, when we see capital B and small b, so we see here actually the capital B should make to produce large starch grains, but instead we see here formation of intermediate starch grains, which means they are not larger starch grains or smaller starch grains, they are of intermediate size. And but when coming to the shape of the seed, it is round. So when coming to shape of the seed, it is round, right? So here when we see from the point of starch grain size, so when we see from the point of starch grain size, okay, here this one is showing incomplete dominance, okay, showing incomplete dominates. But when coming to the shape of the seed, when we consider, so this one is actually showing from the point of shape of the seed, it is showing, okay, complete dominance, is showing complete dominance. So here in this situation we can see that a gene product is producing more than one, okay, phenotypic effect. So actually one gene produces one phenotypic, okay, one phenotype. So it produces only one phenotype. So one gene produces one phenotype, okay. So here so let's say one gene, so it produces, okay, a product, and that will be, okay, expressing as one phenotype, as one phenotype. Okay, so I think this is quite difficult to understand at present. So what is this gene, the product, and one phenotype? This I can explain only in the next chapter, molecular basis of inheritance. So we have still questions that what is gene, in what form a gene is present, okay, and how this gene is expressed. This is what we call expression of a gene. So all those things we will slowly understand, okay, in our coming sessions. So one gene, it produces the product, okay, and that will show one phenotype. But in this case one gene is showing, okay, more than one phenotypic effect. So you can see at one point it's showing incomplete dominance, and from another point, okay, it's showing complete dominance, right? So that's, I'll write a single gene product, a single gene product in some situations, not in all cases. So in, okay, a single gene product may produce more than one effect. Okay, so a single gene product may produce more than one effect. Okay, so that is what we see in this starch synthesis in pea seeds, and this makes us little difficult to understand what actually is dominance and what is recessive. Of course, in the last session we tried to understand, okay, how a particular gene or allele becomes dominant, and okay, the other one will become recessive. So now, okay, in after studying this we will understand, right? So with this we understand that the dominance is not an autonomous feature of a gene. So it means the dominance not alone depends on the gene. Okay, so here the dominance not alone depends on gene. Okay, so that's, the dominance is not an autonomous feature of the gene, right? So here we understand that it depends on the gene product. So here the dominance depends on gene product. So I already told you that, okay, the gene will get expressed and produce a product, and that will show, okay, its effect, that is a phenotypic effect, right? So the dominance depends on the right gene product, and also it depends on the particular phenotype, okay, that is produced on this gene product, right? So that's dominance depends on gene product, okay, and also, okay, the phenotype expressed from this gene product. Okay, so the dominance will depend on the gene product, and it also depends on that particular, okay, phenotype which is produced from this gene product. So both of them will contribute to the dominance of a right right allele, right? And this is in case more than one phenotype, okay, is affected by single gene. Okay, so this will be in the case, okay, more than one phenotype is, okay, or I'll say is influenced by single gene. Okay, so that's all about the starch synthesis in pea seeds. So with this, I will end today's session. So we'll see in the next session chromosomal theory of inheritance, and also linkage and recombination.